Actually, when I came over, my wife and I came for 2 years. So we left all our stuff behind in England, our bed, my records, everything. And we never went back. And we've been here for 30 years now, in Little Rock for 16. And we really enjoy it here very much. It's great. Anyway, I'm a basic scientist, so I don't work with patients. I work in a laboratory and in an office. And we are studying the basic mechanisms of action of anti-tumor agents. As you know, cancer is treated mainly through surgery, radiation, and chemotherapy. And we're studying the drugs that are used to treat cancer. Now, it may come as a surprise that we don't know how these drugs work. But the fact is, we really don't on a detailed level. So typically, the way anti-cancer drugs are identified is through screening. So you have cancer cell lines, or you have tumor models, animal models, and various compound libraries are sought and tested. And something is found that works, it kills cancer cells in the lab or in an animal. And then there's further developing of that compound, and eventually it gets into the clinic, etc. So empirically, we know these things work, but in fact, when you get down to the cellular level, we really don't understand how they're working. So that's really the kind of area I'm in right now. And it turns out that a lot of these anti-cancer drugs, the way they actually work, they get into the cell and they cause some kind of damage. A lot of drugs that are used damage the DNA, which, as you know, is the chromosomes, your genetic material. A key drug in lung cancer, for example, is cisplatin. And it binds to the DNA and damages it. Now, the cell recognizes that there's something wrong. It can't continue dividing because there's a perturbation in the DNA template. There's another drug that's often used in lung cancer that's called Taxol. It's also used in breast and ovarian cancer. What that drug does, it basically blocks cell division at the stage of mitosis. So this is the stage where one cell is dividing into two cells. So basically the cells figure out that something's wrong, and then they kill themselves. They commit suicide. So the cell actually goes into a program of cell death because it's recognized that it's damaged and it can't continue dividing anymore. So some of these mechanisms, some of these pathways that result from DNA damage to cell death or mitotic perturbation to cell death, that's what we're trying to figure out. And it turns out that these pathways are actually pretty complicated. There's a lot of proteins involved, a lot of enzymes involved, and often cancer cells actually become resistant to these drugs because they're genetically unstable. So you may ask, well, who cares? If these drugs actually kill the cancer cell, who cares how they work? Well, we care for a number of reasons. One is that many cells actually become resistant to these drugs. So we want to know why. Why are some cancers resistant and some are sensitive? Can we actually screen patients to figure out which drugs are going to be the most effective drugs? Secondarily, by studying these pathways, we can actually uncover new targets. So we can actually start making chemotherapy a little bit more selective by saying, let's go after this protein or that protein, rather than just use a drug which kills cells through division, killing division, which is also very toxic to normal cells. So actually the drugs like Taxol and vinblastine, these are microtubule inhibitors, they're the ones that we're studying in some detail right now.
Thank you.